please. Thanks, Diego, and thanks everybody again for the invitation of organizing this. I'm very happy to present once more some results on the brain and the stomach. This time in a large cohort of healthy participants as part of a project with Leah Vanelis and Micah Allen. So one of the motivations, it still seems to be the same motivation after all this year, is trying to understand what is the reason of the brain always producing what we call intrinsic activity, right? The brain is always active, and we tend to think that it's associated with perception of the external world, cognition. But as we know, all the people that are here, that the brain is always trying to keep the body alive. So the rhythmic visceral signals, such as the heart, respiration, or the stomach, could be a source of influence for this. And now more than ever, I think that the anatomical pathways that connect the viscera to the brain are more and more important to understand this coupling. And what I'm showing you here is a summary of what in an animal model will be the regions that receive ascending inputs from the stomach. And you can see that it has quite a number of important regions like neuromodulatory structures, serotoninergic, acetylcholinergic, noradrenalergic, as well as behavioral effectors like the amygdala, the hypothalamus, the hippocampus that actually drive behavior. And I think they have the potential to subconsciously influence mental life. So the stomach in particular produces pacemaker at the frequency of the resistant networks that we measure with fMRI. And one of the main motivations of my PhD project, that now is still doing it after 4 years of postdoc, is trying to understand which are the brain regions coupled to the stomach and why. So the startup point of this was this paper we published in eLife now 4 years ago, trying to see which bold regions, which fMRI regions were coupled with the stomach, and we found it was mostly visual and somatomotor regions, and also regions that before our colleague Craig Richard found with the alpha rhythm using MEG. Now, for those of you who don't know, were not there yesterday, we can record the signal from the stomach using electrogastrogram, so electrodes on the belly, like an electrocardiogram, but much smaller amplitude, of course. And the signature is this peak at 0.05 hertz, that if we filter, we can recover the slow rhythm of the stomach, and on top of that, the faster rhythm of respiration and of the heart that we can filter out, as well as the scanner noise shows by filtering. And for this study, what I mostly use is the phase of the electrogastrogram to see basically when the stomach is in a more expanded or contracted state, even actually without contractions. So we have 3 follow-ups. There were 3 follow-up studies in humans after this. My own work with Catherine Talon-Boudry, collaborators with Niels Kromer and Sophie Müller, and also a team in Israel by Gail Avidan and colleagues, that they also replicated this study, this gastric network, the fact that these brain regions coupled to the stomach, but it was only with 60, 50, or for 30 subjects. So now we wanted to do this with 243 subjects with clean data out of 300 subjects that actually had complete data, that they went through a resting state fMRI session with eyes open. We have the electrogastrogram, of course, and what we did, the main method was to quantify the phase coupling between these two. So in a nutshell, you will see in blue the trace of the stomach, and in orange the trace of the brain. This is in a brain region that is coupled, where the time delays of the 2 signals is consistent. This is another brain region. So we try to quantify this at the group level, and then find how is it related to which are at the group level. We compare them using surrogates by shifting in time the ECG, and we can make a t-test and correct for multiple comparisons. And we try to characterize the overlap with resistant networks, with the previous studies that I mentioned before, and with neuromodulatory systems of group level PET data. So our group level results with a t-value, so using the same threshold that I used before with 30 or 60 participants, I basically found that the whole brain is coupled to the stomach, which was, okay, this might be a little much, but it does speak that potentially the gastric coupling is pervasive across the cortex. Now we use a much more stringent threshold. We found basically the same regions that I found before, that are basically sensory motor network and a little bit of the default and the saliency network, and to some degree the visual regions. So I'm surprised how come the visual cortex is coupled with the stomach, where there are multiple pathways that can connect the stomach with the visual cortex, believe it or not. Happy to talk about that later on. And effect sizes were largest in the sensory motor regions, which makes sense in the sense that these are the regions that likely control the stomach. So the largest coupling indeed in sensory motor network, and we found some very nice overlap with one of my first discoveries of the gastric network, the replication with the team of Niels Kromer, and also with an independent lab that is using completely new data. So this made me very happy because actually this phenomenon is replicating across studies. I think nothing makes a researcher more happy than replication and funding. And I think what is the function now? Now we can see that this is actually stable across multiple data sets. Well, I do think the function is really much related to a neuroanatomy. And this is the first trial we're doing of trying to explore this new, very rich data set that we have Leah with Micah. And what we do is, for this particular day, is try to compare the spatial map of this gastric network with libraries that we have and part of this neuromap software about, in this case, neurotransmitters distribution systems. For example, the neurotransmitter noradrenaline, where in the brain is dense or not. So what we do is we compare a correlation analysis of which voxels are both part of the gastric network and have also large density of noradrenaline, and then we shift in space the map. We made the brain a sphere, we shifted that it was a complete sphere, and then we tried to see how much overlap you could expect by chance if the spatial distribution was the same but shifted across the cortex. So we can, this way, get a p-value for an overlap with each of these neurotransmitter maps. And I'll go only for the significant ones. So I tried to compare with all of them that were available to see, okay, what is going on here? And surprisingly, we get the serotonin transporter number 6, noradrenaline transporter, acetylcholinergic system, glutamate, also an excitatory neurotransmitters, synaptic density, so regions that have lower synaptic density and less energy efficiency. So I don't know if you start seeing, but the picture here is somehow related to both arousal, so brain neuromodulatory system that makes the brain awake or sleepy, and also of how efficient the transmission of the signal is. And of course, if you come back to this first picture here, what it's showing is that the brain regions that we do see quite all of the cortex, but we're also seeing the regions that can actually modulate all of the cortex. So in a way, it makes sense. So yeah, brain-stomach coupling could be a mechanism to regulate the brain energy metabolism as a function of arousal. And I have a hint of a potential other function that may be not only the brain energy metabolism, but also the whole body energy metabolism. This is a study by the amazing team in Israel, Levakov, from Galia Vidal. They have 92 participants that went through a weight loss intervention. They were eating only 800 calories for like 3 months, and they used a decoder to find out which were the regions that predicted weight loss, and these were there in blue. And they compared the overlap with our gastric network, and indeed they overlap. And they went further to show that the amount of power that patients with obesity had at 0.05 hertz, the frequency of the stomach, was predictive of how much weight they will lose. So that people that have more power at the frequency of the stomach will lose less weight. Also, if they shifted the filter for higher and slower frequencies, this correlation will disappear. So it seems that this gastric coupling could eventually not only be related to brain energy metabolism, but the metabolism of the whole organism. Yes, it remains to be function. My own interpretation of this is that this brain-stomach interaction is related to arousal, metabolism, and probably also affective experience, which are all in a way directly influencing mental life. And at the end, I think it's all in the anatomy. Thank you very much for your attention. I'd like to thank Leah and Mike, who have been excellent support in all this project, and hopefully much more to come. Thanks again.
So do you have questions in the public? No? Anyone?
Thanks a lot. A very general question. What is the function of this gastric rhythm?
What's
the function of this gastric rhythm?
This gastric rhythm is actually pacing the contractions of digestion. So the stomach contracts every 20 seconds, and when it does contract, it contracts at this frequency. So we need it to actually make the food goes down.
Okay,
but
so
even when there's no digestion, still you have this rhythm going on.
But it's related to how much food do we take and which moment of the day and how do you
Actually, so far we have tried to make many contours analysis, and it seems to be quite independent of the speed of the stomach, of what did you eat. Maybe if you are hungry or not, we haven't really tested if you're really hungry, but if the stomach goes slightly faster or not, if it's having a lot of contractions, it seems to be independent of how much coupling actually you have.
Thanks.
There was another question.
Thank you very much for the wonderful talk. It's a bit yeah, I don't know, not off topic, but maybe standing in, I don't know whether you have an answer to it, but I was wondering whether you know of any study that would, for example, look at CSF flow and whether it's somehow linked to the gastric cycle that you presented. 'Cause there's also fluctuations in a similar time scale, if I remember correctly.
No, there's not off topic at all. Actually, in human memory mapping, there was a colleague, I cannot remember her last name, but they were actually starting to look into that and trying to see if this is one of the modulators, especially during sleep. They started to have a CSF flow, and they were pretty interested in looking at if the stomach is actually shifting this, making the CSF go a different direction. It could be, yeah.
Okay, great. Thank you.
Thanks.
I do have a question for you. What is your intuition that why you repeatedly find that it's coupled with the sensory motor cortices to your gastric rhythm? And then why people are trying to link the gut with things that are mostly frontal, like decision, metacognition, and so.
Well, that's a good point.
Like it doesn't fit the whole story, you know.
Yeah, that is true. Look, I think my take is that I mentioned them during resting state, and I think Daniel have something showing that maybe not the same when doing some kind of task. So I think it's the sensory motor regions because first, these are the ones that most densely receive spinal inputs from the stomach and also vagal inputs from the stomach. So sensory motor cortex is actually sending and receiving inputs, and is in a way it's part of the sensorium. So even if you are not consciously perceiving the stomach, your sensory cortex is also processing it. So I think that's my guess. Maybe if you have to make a food decision-making task, your stomach will actually be coupled to the frontal cortex. I don't know, but I have no idea.
Okay, thank you. Sorry. Last question.
Thank you for your talk. And I was wondering, there's a DAS value. You mentioned that. I can see there's a corresponding p-value. Could you tell me what the p-value refers to?
Here?
Please.
Here, yeah, this is so this comes to here. For all of these neurotransmitters, we have a p-value, and this p-value is the chance of having an overlap this big, given that it is not true. So the way we obtain this p-value is here we have an empirical overlap between the gastric network and each neurotransmitter, and then we shift it across the whole cortex, we wrap it around as a sphere, and then we put it back into a real brain. We do this 1,000 or 10,000 times, and the p-value is how many of these random surrogates overlaps is larger than the actual real one.
Roger. It's Kanda permutation.
Yeah, it's a Monte Carlo permutation, yes.
Okay. Thank you, Ignacio.